Hey, how's it going, everyone? This is FixerMed. Welcome back to my high-yield immunology review series for the USMLE Step 1, NBME CBSE, and NBME CAS examinations. This will be part 5 of my multi-part video series covering the entire discipline of immunology in a high-yield but comprehensive manner. So without further ado, let's go ahead and get started on today's content. So I'd like to open up today by talking about the differentiation factors between CD4 plus T cells. I'd like to explore CD4 plus T cell differentiation and focus on the Th1 and Th2 pathways and how CD8 plus T cells are primarily activated for target cell destruction. Some key takeaways are that Th0 can differentiate into Th1, Th2, Th17, and T regulatory cells, and Th1 cells activate macrophages. Th2 cells simulate B cell antibody production. The picture on the right should illustrate this a little better, but let's go ahead and move on to the next set of content. So let's go ahead and narrow in and focus on the Th1 differentiation pathway. The main trigger for this is stimulation with the IL-12 and interferon gamma. IL-12 from macrophages initiates differentiation. Interferon gamma further activates macrophages. Importance here is the IL-12 interferon gamma loop critical for Th0 to Th1 transition. And the key outcome here is macrophage activation, potential formation of granulomas, and chronic inflammation. Let's go ahead and move on to the next set of content, which is discussing Th2 differentiation pathway and interactions. So the main trigger here is stimulation with IL-4 and IL-10, and then we have CD40L and CD40 interaction, which facilitates B cell activation and antibody production. Then we have Th1, Th2 interactions, IL-12 interferon gamma, which suppress Th2 lineage, and IL-4, IL-10 suppress Th1 lineage. So the key outcome is that somatic hypermutation and affinity maturation enhance antibody specificity. And a visual representation can be found in the pictures to the right on all 3 slides. That's how I learned it, just drawing it out over and over again. So if you still don't get it, let's go ahead and try to do some practice problems now. Which of the following accurately describes the differentiation of Th0 cells into Th1 and Th2 cells? Alright, I think I gave you guys enough time. If you need more time, please feel free to pause the video. Otherwise, moving on now. Th0 cells differentiate into Th1 cells via stimulation via IL-12 and interferon gamma. This is supported by the understanding that IL-12 from macrophages initiates the differentiation process, and interferon gamma further activates macrophages, forming a critical loop for Th0 to Th1 transition. Th1 cells are characterized by their ability to stimulate macrophages, leading to phagocytosis of pathogens and potential granuloma formation and chronic inflammation. Therefore, the correct answer is B. Th0 cells differentiate into Th1 cells via stimulation via IL-12 and interferon gamma. Let's go ahead and see why the other answer choices are incorrect. Incorrect answer choices can be explained as follows. Th0 cells differentiate into Th1 cells, not Th2 cells, and this process is mediated by IL-12 and interferon gamma, not IL-4 and IL-10. C. Th0 cells differentiate into Th1 cells, not Th2 cells, via stimulation with IL-12 and interferon gamma. Th0 cells differentiate into Th2 cells via stimulation with IL-4 and IL-10, not Th1 cells. IL-6 and TNF-alpha are not primarily involved in the differentiation of Th0 cells into Th1 or Th2 cells. Let's go ahead and move on to the next question. Which of the following accurately describes the differentiation of CD4 plus T cells? Alright, I think I gave you guys enough time here. If you need more time, please feel free to pause the video. Otherwise, moving on now. CD4 plus T cells can differentiate into Th1, Th2, Th17, and T regulatory cells, each with distinct functions. This aligns with the understanding that CD4 plus T cells have the capacity to undergo various routes of differentiation, resulting in different T cell subsets with specialized roles in immune response modulation. Therefore, the correct answer is C. CD4 plus T cells can differentiate into Th1, Th2, Th17, and T regulatory cells, each with distinct functions. Let's go ahead and see why the other answer choices are incorrect. Incorrect choices can be explained as follows. Th1 cells primarily stimulate macrophages, while Th2 cells are responsible for activating B cells for antibody production. CD4 plus T cells do not directly kill target cells, but instead regulate immune responses through differentiation into various T cell subsets. Th1 cells do not suppress autoimmunity by inhibiting T regulatory cell activity. Rather, T regulatory cells play a role in preventing autoimmunity. Th2 cells do not primarily stimulate macrophages for pathogen destruction. They mainly stimulate B cells for antibody production. Let's go ahead and move on to the next question. Which cytokines play a crucial role of differentiation of Th0 cells into Th1 cells? Alright, I think I gave you guys enough time here. If you need more time, please feel free to pause the video. Otherwise, moving on now. IL-12 and interferon gamma are crucial cytokines in the differentiation of Th0 cells into Th1 cells. IL-12 secreted by macrophages initiates the differentiation process, while interferon gamma further activates macrophages and contributes to Th1 cell development. This critical loop of IL-12 interferon gamma is essential for the transition of Th0 cells into Th1 cells, which are known for their role in stimulating macrophages and orchestrating cell-mediated immunity. Therefore, the correct answer is C. IL-12 and interferon gamma are crucial cytokines in the differentiation of Th0 cells into Th1 cells. Let's go ahead and see why the other answer choices are incorrect. IL-4 and IL-10 are associated with the differentiation of Th0 cells into Th2 cells, not Th1 cells. IL-6 and TNF-alpha are not primarily involved in the differentiation of Th0 cells into Th1 cells. They are associated with other immune responses. IL-2 and IL-17 are not key cytokines in the differentiation of Th0 cells into Th1 cells. IL-2 is involved in T cell proliferation, while IL-17 is associated with Th17 cell differentiation. IL-1B and TGF-beta are not primarily involved in the differentiation of Th0 cells into Th1 cells. They have roles in other immune responses such as inflammation and regulatory T cell function. Let's go ahead and move on to the next question. Which cytokines are involved in the differentiation of Th0 cells into Th2 cells? Alright, I think I gave you guys enough time here. Pretty first order questions today, so let's go ahead and move on and see what the correct answer choice is. If you need more time, please feel free to pause the video, otherwise moving on now. Th0 cells differentiate into Th2 cells via stimulation with IL-4 and IL-10. IL-4 plays a crucial role in promoting Th2 differentiation and facilitating B cell activation, while IL-10 contributes to the anti-inflammatory functions associated with Th2 responses. This pathway leads to the development of Th2 cells, which are primarily responsible for stimulating B cells to produce antibodies and mediating humoral immunity. Therefore, the correct answer is C. Th0 cells differentiate into Th2 cells via stimulation with IL-4 and IL-10. Let's go ahead and look at why the other answer choices are incorrect. IL-12 and interferon gamma are associated with the differentiation of Th0 cells into Th1 cells, not Th2 cells. IL-6 and TNF-alpha are not primarily involved in the differentiation of Th0 cells into Th2 cells. They are associated with other immune responses. IL-2 and IL-17 are not key cytokines in the differentiation of Th0 cells into Th2 cells. IL-2 is involved in T cell proliferation, while IL-17 is associated with Th17 cell differentiation. IL-1 beta and TGF beta are not primarily involved in the differentiation of Th0 cells into Th2 cells. They have other roles in other immune responses such as inflammation and regulatory T cell function. That will do it all for content today, guys. If you have any questions, comments, or concerns, please feel free to leave them in the comment section below. If you like this type of video, please be sure to like and subscribe for more content like this. Otherwise, this is FixerMed signing off. Be sure to have a great day, everyone, and good luck studying. Goodbye.